Okay, hi everyone. So welcome back to another session. So this time our session is going to be on the identification and documentation of macrofungi. So since the rainy seasons are nearby, and this time I guess the mushrooms have already started coming up, and let's have this session to understand the world of mushrooms and a better way to how to document it. So today we have Harikrishnan M T as a speaker. So he's a dedicated microbiologist and a mycologist. He's a founding member of Mushrooms of India and Shroom Sabha Foundation. And he's like very active in this. So he has even co-led the Kerala's first macrofungal survey. And he even has authored a beginner's guide on Bangalore's fungi landscape. And you must have seen him in various of the fungi groups. So he's one of the most active ideas who give IDs and promote the love to fungi. So over to you, Hari Krishna.
Hi, good evening, everyone. I know it's very late. Uh, yeah, let's not waste much time. Yeah, so thank you, Ashwin, for the intro. Uh, I I have been active in this space, and I have been active in identification of mushrooms, basically. So that's what I do apart from my career. So yeah, so today's session we'll be learning little about identification of mushrooms and okay. So we'll be learning little about the identification of mushrooms, the macrofungal or macromorphological characteristics of this particular mushrooms. So we'll be learning few important macromorphological characteristics that will aid in identification. Also, we'll be talking a little about tips for identification or tips for documentation of mushrooms. So as to get a proper ID for your mushroom pictures, you have to know how to document those particular mushrooms. So how to effectively or efficiently document a mushroom, so that is what something we'll be learning about today. And we'll learn or we'll be discussing little about few points to note while uploading mushroom observations in iNaturalist. Uh, so as I've been IDing mushrooms in iNaturalist under the name Indian Toadstool. So one issue we always face is the quality of observations. So there will be insufficient images, and such things which are important for identification. So we'll be discussing few of those things in this uh, particular portion, that is few points to note while uploading the mushrooms observations in iNaturalist. Okay. So first of all, what is mycology? So mycology is basically the branch of science that deals with study of fungi. So myco is a term that relates to fungi. So wherever the term myco comes in, it is something that is related to fungus. Okay, so whoever learns about or studies a particular fungus is called as a mycologist. So as to learn about mushrooms or fungi, you have to understand certain characteristics that this particular group of organisms can possess. So the first and foremost, so there are certain characteristics for an organism to be classified as a fungus. So just like how we humans have certain characteristics uh, that makes us eligible to be under say kingdom Animalia, right? So in that sense, there are certain characteristics that fungus should possess. Okay, so the first and foremost characteristics, so these are some of these are in the cellular level. So uh, although mushrooms you can see with your naked eyes, fungi in general are considered as microscopic organisms. Okay, so they are microscopic organisms. So in their cellular level they have certain characteristics. So the first and foremost characteristic is eukaryotic type of cell. So there are two different types of cells, the prokaryotic cells and eukaryotic cells. Prokaryotic cells are Primitive cells and eukaryotic cells are more advanced type of cells. Now, prokaryotic cells usually you will find in organisms like bacteria. Uh, so, eukaryotic cells you will find in fungi, plants, and animals. So, this is the in a cellular level. Now, these particular cells, these particular eukaryotic cells, have a chitinous cell wall. So, chitin, can anyone tell me what is chitin? You can unmute.
Could I see the answer?
The question is what is chitin? You can
Okay.
yeah.
So chitin is basically I think hard protein of sorts that is found not only in the I think exoskeletons of most insects, but also in fungi. I think I'm not very sure.
Yes, yes. So chitin is basically a structural component. Okay, so it's a structural component which gives rigidity to the cells. So this particular fungi so have chitinous cell wall. So in case of plants, they have what kind of cell wall? What kind of cell wall does a plant have?
Am I to answer the question?
Anyone can answer. Yes,
Cellulose.
yes, cellulose. Yes. So these are uh, basically macromolecules. Yes, it's a polysaccharide. So these are components that give structural rigidity or structural strength to the cell. So in case of fungi, they have chitinous cell wall. So earlier, researchers used to classify fungi under kingdom Plantae. So later, when we started looking at these particular organisms, we understood that they have certain characteristics that sets them apart from. Okay, oh, my video is off. Just a second. Uh, can you see? Yeah. So they have certain characteristics that sets them apart from plants, right? So one of those characteristics is a chitinous cell wall. So they have a chitinous cell wall. They are non-vascular. So in case of plants, they have a vascular system. They have a well-defined vascular system, which is the xylem and phloem, right? In case of humans, we also have certain circular system. So we have certain pathways or certain tubular structures through which certain materials are transported. So vascular system is basically some tube-like structures through which the components or through which material is transported. So in case of humans, we have blood vessels for transport of what is it for?
I think circulatory fluid or blood.
Yes. So it is ultimately transport of oxygen, right? So the blood cells or the RBCs will carry the uh, oxygen. So oxygen or carbon dioxide, whatever it is. So respiratory gases. So same way, plants also have. In case of fungi, we don't or they don't have a vascular system. So they are non-vascular. So instead of having tubular structures, the components that are to be transported are present in the cytoplasm or they are freely flowing in the cytoplasm. One second. Yeah. The next characteristic is they are non-motile. So they don't have specialized structures for moving from one point to another. So that is why they are called as non-motile. They are achlorophyllous. Again, another characteristic that sets them apart from plants is that they lack chlorophyll. So, what is chlorophyll used for? Yes. So, chlorophyll is used for photosynthesis. So, basically, they produce their own food, right? So, they prepare their own food. So, in case of fungi, they don't have the ability to prepare their own food. So, in such cases, what they'll do? So, in case of fungi, how do they consume or how do they? So, what is the mode of nutrition for fungi? So it is saprophytic, right? So or saprotrophic. So that is the mode of nutrition wherein they don't prepare their own food. They feed on dead and decaying plant matter. So there are other modes of nutrition also that fungi. Yes, yes, they also take up parasitic mode of nutrition. So there are other forms or other modes of nutrition, but predominantly it is saprophytic mode of nutrition. Then they are either unicellular or filamentous organisms. So in case of say animals, right? So higher animals. So you have in case of humans, we are multicellular organisms, but fungi are usually unicellular or filamentous organisms. Can anyone give an example of a unicellular fungi that we usually use, or it is
Yeast.
usually a part of
Yeast.
our life? Yes, yeast is a unicellular fungus that we commonly consume and use in our day-to-day life. So, if an organism possesses all these characteristics, you can classify them under kingdom fungi. Yeah. So let's briefly see the classification of kingdom fungi. So this is not something you have to by heart or remember. So this is just to understand that this particular kingdom is very diverse. So this is from the so this is one of the latest uh, classification system. Okay, sorry, latest classification of fungi. So in this, you can see I have given a pie chart wherein you can see majority of these fungi fall under two phyla, right? So that is Basidiomycota and Ascomycota. So most of the mushroom forming organisms or fungi falls under this particular group, that is the Basidiomycota. So Basidiomycota is where most of these organisms or most of these uh, so most of these mushroom forming organisms are found. Some of these are also found in Ascomycota, phylum Ascomycota. Now, this is just for your understanding, but it is not very important for our next session, the next slides. Okay. Yes, yes, you can. Yeah. So if you have any questions, you can post it in the chat. So I will check the chat at regular intervals so that we'll be able to cover the questions or the
Uh,
doubts.
so could I ask my question right
Yeah,
now?
yeah, sure, sure.
So I've never really understood the difference between slime molds and fungi and protozoans, since some slime molds seem to have the um qualities of both, you could say. For example, they're unicellular, they can move
Yes.
around, but they also and they're amorphous like amoebae, but they also behave exactly like fungi in that they have fruiting bodies and spores, etc. So I never really understood the difference. If you could.
So they have alternating what do you say a type of morphology. So uh, they attain a amoeboid form of morphology, and so that's there are two stages in their life cycle. So for one is the amoeboid form, and then is a sporulating form, which is the mushrooms, right? So in case of fungi, you don't usually find this particular type of dimorphism, or two different sexual morphs. So that is not usually found in fungi. So that is a major difference. So again, they fall under another kingdom. So they fall under protozoans. So they fall under protista. So
So,
that is the yeah.
so are you implying that the the different life forms depend on the gender of said um slime molds?
I didn't understand the
So you were answering, you said that they had two different genders of sorts. So are you implying
yeah.
that each form is for each gender? Like, for example, the female gender might have
No, no,
fruiting
no,
body.
no, no. Sorry, sorry. It's not. It's not sexual dimorphism. So Basically what happens is that I don't think they have such a like, I don't know if they have sexual dimorphism. I am not aware about that. But usually what happens is that the life cycle of this particular organism have two stages. So in initial stage, they'll possess the amoeboid form, and then so sporulating forms are usually formed when there are adverse conditions, because spores are very hardy cells, so they can survive under unfavorable conditions. So that particular switch from amoeboid form to a sporulating form is found in this particular slime molds. So earlier it was classified under fungi, but then it was moved, or it was removed from fungi and was given a separate, or uh, was placed under protista. So because of their even the cellular structure is different. I believe it is different. Uh, so yeah,
Thank you, sir.
yeah. Okay. So what are mushrooms? So we spoke about fungi, right? So we spoke about what are the characteristics of a fungus. And now there are different types of fungi. There are yeast, which are unicellular, which do not form mushrooms. So what are mushrooms is a question that most people uh, so it's a doubt that most people have. So mushrooms are basically the fruiting bodies. So they are comparable to fruits. So when you take a tree or a plant, right? So what is the function of a fruit? Anyone can answer.
To spread more seeds of the said plant.
Okay. So what is that function? Yes, propagation. So they are reproductive in function, right? So similarly, mushrooms are basically the fruits of certain groups of fungi. So they are fruits. They are not seeds. They are not comparable to seeds. They are comparable to fruits. So fruits usually have seeds. So similar way, mushrooms have spores. So there are fungi. So you can compare the fungal mycelium. So this particular white body or white mass, thread or filamentous mass that you see is called as the mycelium. So they usually have mycelium. So mycelium is comparable to the tree, and mushrooms are comparable to the fruits, and spores are comparable to the seeds. Now why is this information important? So here, based on this information, we have uh, we have, so this implies that mushrooms have a reproductive function, right? So they are reproductive in function, and the reproductive cells are spores. Okay. Now, this is how usually a what do you say, mushroom looks like. So people usually when they see mushrooms, they think that mushrooms are formed by a different type of cell, which is different from the mycelium. But basically, it is the same type of cell. So mycelium, which are interwoven to form a specialized fruit-like body. That is what mushrooms are. Okay. So that is basically what a mushroom is. So mushrooms are basically specialized bodies, or they are just mycelium which are interwoven. So if you want to uh, say cultivate particular mushroom, if you want to grow a particular mushroom, you can take a tissue from this and you can do, so how you usually do tissue culturing, right? Same way you can culture mushrooms from this particular tissues, because they are composed of mycelium. Okay, so these are mycelium, the thread-like structures. So each thread or each thread-like structure is called as a hyphae. So the whole thing is a mycelium, and each thread-like component is called as a hyphae. Any doubts till now? Okay. So this is the life cycle. So what happens is usually the spores, when they fall on the medium on which they have to grow, what happens? They form hyphae. Okay. Now, in case of mushrooms, can anyone tell me if it is a sexual reproduction or a asexual reproduction?
Asexual reproduction.
Okay, so in case of mushrooms, it is actually sexual reproduction. Okay, so when there are two compatible spores, okay, two compatible spores. So I hope you know the difference between the sexual and asexual mode of reproduction, wherein so the sexual cells will be haploid, meaning that they'll have half the number of chromosomes, and when they mate, it becomes a diploid, having two sets of chromosomes, right? So we humans have two sets of chromosomes, but our sex cells will have half the number of chromosomes. So two sex cells, when they combine together, it forms a diploid, or a body with two, what you say, sets of chromosomes. So similarly, here you can see two hyphae, compatible hyphae, when they come, when the hyphae that is formed by these two compatible spores mate. So this is mating. Okay, so when they mate or when they merge together, so there are certain structures, clamp-like structures that form, that enables them to fuse like this. So once they fuse, what happens? The genetic material get what do you say, remixed, or they get mixed, and they form a diploid mycelium. So the mycelium is formed. The mycelium, when there is, when the favorable conditions arrive, so in case of, so daily we see, right? So whenever there is a rain, or whenever the temperature is uh, very cool, right? So when the temperature is cold and when there is enough moisture, you find mushrooms popping up. So these are favorable conditions for fruiting. So the mycelium, when they get favorable conditions, they form the mushrooms. Now the mushrooms again forms haploid spores. Okay, so this is the basic life cycle of a mushroom, wherein the spores, when two compatible spores mate, they form the hyphae. Sorry, mycelium. The mycelium will form mushrooms when the conditions are favorable. The mushrooms will form spores. Again, the life cycle continues. Now, uh, I told you that mushrooms are comparable to fruits, right? And so, in case of documentation, so there are certain things that we should know. So, in case of documenting mushrooms, at some point you might have to pick the mushroom. Okay, so you might have to pick the mushroom to efficiently document mushrooms. So in that case, does picking mushroom affect the organism, or how does it affect the organism? Can anyone guess how it will affect? Okay. So what happens is that there is a misconception, or there is a tendency to compare mushrooms to plants, right? So picking mushrooms in moderation. So there is something called as ethical foraging, wherein you don't usually go out and pick all the mushrooms that you find. So usually when the mushroom, or the mushroom that you find, is sporulating or is in its mature state, picking the mushroom usually does not affect the organism, but again, you should be very considerate about the ecology. So, yeah, picking the mushroom does not affect the mycelium. Again, the mushrooms come up from the same spot because the mycelium is still present beneath. Okay. Now, just few things that you have to know about the anatomy of a mushroom. So, a mushroom can be divided into three parts. Okay, so first is the cap, then you have the underside of the mushroom, and you have the stipe or the stalk. Okay, so the cap of the mushroom, the underside of the mushroom, and the stalk. Now, the underside of the mushroom is where the spores are formed and released. So I told that mushrooms are fruiting bodies and the spores are the seeds, right? So the seeds or the spores are usually formed on the underside. So there can be different structures on the underside of a mushroom. It's in this case, in this case, the underside of a mushroom is gill. So there are other structures that can be present. Okay, so it is important to divide these structures into three because we'll be getting deeper into these structures. Okay, so we'll be looking at these structures in detail. There are two more things that you should know when you talk about the anatomy of a mushroom. So the first one is a universal veil. So when some mushrooms at their younger stage, so when they are very young, they are usually covered by a membranous structure. So this particular membranous structure protects the young mushroom. Okay, it protects the young mushroom, and this structure is called as a universal veil. So what is a veil? Veil. What is a veil? What is the meaning of a veil? You can, yeah, it's basically a cover, right? So it's basically a protective layer that is present on or over the young mushroom. Now there is something else called as a partial veil. Now partial veil is something that usually present or usually covers the underside of a mushroom. So this is a structure that usually covers or protects the underside of a mushroom. So why is this protection important? Why do we have to protect, or why does the mushroom have to protect the underside of a mushroom? What is present or what is the function of the underside? So it is basically present to produce spores. Yes. So it is a spore bearing surface, or it is the fertile, sorry. Yeah, it is a fertile surface. Okay. So it is a fertile surface. So partial veil is present to protect the fertile surface of the young mushroom. So as and when the mushroom starts maturing, what happens is that these layers are broken. So the universal veil can break, and part of it can remain at the base or at the top. Okay. So it can either remain on the top or at the base or both. Okay. So it can remain at the base or top or both base and top. And also it could be lost. Okay, so in some species, what happens? It will only be present on top of the mushroom. In some species, it will only be present at the base of the mushroom. And in some species, it can be present on top as well as base. And in some species, it could get lost. Okay. Then again, the partial veil, which was protecting the underside, right? Again, when the mushroom starts opening up, so when it starts opening what will happen? This particular layer can get removed. Okay, it breaks. So again, here also it can either be or it can either remain on the stipe or it can remain on the margin of the cap. So this is how it was attached, right? From the stipe to the margin of the cap. So it can either remain on the stipe or on the margin of the cap, or it could be lost. Okay. Now, these structures are important for identification of mushrooms because these are structures which are specific to certain genera or certain species. So, presence or absence of these structures help in identification. Any doubt? I think someone was raising their hand. So, you can unmute and ask if you have any doubt. Okay. Next is tools for identification. So now we have discussed about the anatomy of a mushroom or certain parts of mushrooms which could help in identification. Now there are three major tools for identification of mushrooms. The first one is macromorphology. So whenever uh, we try to identify an organism, we usually check what are the characteristics that are visible to us, right? So visible to our naked eyes. So, say humans, we have certain macromorphological characteristics. We have two hands, we have two legs, we walk upright. Okay, we have certain hair structures, right? We have facial hair, we could have facial hair. We have two eyes. So, these are certain macromorphological characteristics. That is, the characteristics that are perceivable to our naked eye, right? Then there are microscopic characteristics. So, in the beginning of the session, I said that mushrooms are or fungi are microscopic organisms. So, there are many things that happen in a cellular level. Okay, so the spores, there are spores of different sizes, different colors. Then the types of cells that are present in the on the underside, on the top of the cap. So these are so presence or absence of these particular cells. All these information comes under microscopy. Okay. Then we have something called as DNA sequencing. So wherein we extract or we isolate the DNA from the particular organism and we read the codons or we read the genetic information that is present in this particular organism. So these are three basic tools that we have. So earlier times when the mycology was in its like very early stage, in its very young stage, we only had this macromorphological characteristics. So earlier when you see very old literatures or very old books, most of the mushrooms were classified into or under a single genus. Okay, so there were only few genera. When microscopy got introduced into science or into biology, or use of lenses got introduced into biology, we started observing these particular organisms under microscope, and we uh, understood that these organisms are different in a microscopic level. So based on the microscopic differences, again the taxonomy got revised, right? The taxonomy of fungus or fungi, it started evolving. Later when we uh, discovered DNA, after discovering DNA, after getting to know that certain, or what you say, there are similarities and differences in DNA, and this similarities and differences in the DNA directly contribute to similarities and differences in our appearances, right? So the genotype influences the phenotype. So we started identifying mushrooms based on the DNA sequences. Okay. So two closely related genera will have more similarity in their DNA sequences than two distant uh, genera. And based on these similarities and differences, we are able to detect something called as a phylogenetic tree or the ancestral relatedness. Okay, how it was related during the course of evolution. So these are the three tools, and the most accessible or available tool for a normal person or a layman is something called as a macromorphology. So based on how they look different, right? Based on that, we identify them. So here these are there are around 15 macromorphological characteristics. We'll not be diving deep into all these characteristics. We'll check few very important characteristic that you have to document. Now, first and foremost is a substrate. Can anyone guess what a substrate is? Any idea? What is a substrate? Okay, there is a question. Does commercial growth of various types of mushrooms, these days we get many types in India, affect ecology in any way? So, yes, in some cases. So, there are some mushrooms that can be invasive. So, there is something called as a Pleurotus citrinopiliatus. So, that is the golden oyster. So, golden oyster. So, earlier in the North American, I think it was in North America. So the cultivators started cultivating this particular mushrooms, and it somehow the spores got out of the cultivation facilities and into the wild, and this was found to be what do you say? Uh, so yeah, it started growing wild, or it was a wild strain, but the cultivar or the cultivable strains of this particular mushroom started overtaking the other uh, natural fungi that were present. So yeah, this can get out of control. So that is why it should be in moderation and under control. Yeah. So substrate. Substrate is a medium on which something grows. Yes. So substrate is basically a medium on which something grows. So in case of mushrooms, substrate is their food. Okay. Substrate is their food. So mushrooms, as I said, they are non-motile. They can't go from one place to another. Right. They can't go from one place to another. uh, and they can't prepare their own food. So what they do is, instead of going from one place to another, what they do is they grow on top of their food. Okay, so that particular food is called as substrate. So substrate is a medium on which the mushroom grows, and in turn it degrades or it uses that particular medium as their food. Okay. Each species or each genera or each genus of mushroom have a preferred substrate. Okay, so the understanding the substrate could lead us to a potential ID. Okay, so when I say pleurotus, I said right, oyster mushrooms. Oyster mushrooms usually grow on wood, right? So they grow on either live or dead and decaying plant matter or wood. So they can't grow in a fertile soil. Okay, so there are certain substrates or there are certain preferred food for these particular species of fungi. So these are some major substrates that mushrooms usually utilize. The first one is decomposing wood. Okay, decomposing wood, wood chips, leaf litter, hay. So basically decomposing plant matter. Then there is something called as live tree. Okay, so mushrooms can grow on live tree. So this mode of nutrition can either be parasitic, wherein it parasitizes the live tree, slowly draining its nutrients and slowly killing the particular tree, or it can be again decomposing, right? It can decompose the live tree. Instead of draining the nutrients, it will degrade the outer bark. It could be the bark of the tree. Okay, so whatever it can degrade, it will degrade instead of what do you say, taking up the already degraded nutrients. Then there is something called as a, okay, so next one is fertile soil. So there are certain mushrooms that grow on manure. Okay, so they grow on fertile soil. So the button mushrooms that you usually consume is a very good example of mushroom that can grow on fertile soil. There is a question. Does reishi mushroom cultivation also affect the ecology? If so, how can we control that? Okay, so reishi or Ganoderma is a very, what do you say? It is a mushroom that sporulates too much. Okay, so it sporulates at a very high rate. And I think it is important to uh, have an enclosement. Okay, have a place which is closed so that the spores are not sent out uncontrollably, because again we talked about this live tree and parasitic mode. So Ganoderma parasitizes live trees. They can kill the live tree, but this particular process is very small, very slow. So it takes a lot of time for the Ganoderma to kill a tree effectively. So it will take years. Okay, so again, it should be under control. Whatever cultivation that you do should always be under control. It should be in controlled environments, under controlled conditions. How accurate is the representation of fungi in Last of Us? Will we be okay? So I'll talk about this in some time. So yeah, fertile soil, best example is button mushrooms. Then there is something called as coprophilous fungi, fungi that grow on dung. Okay, so whenever you go into the wild, when you find, say, an elephant dung, or uh, what you say, wild cattle dung, or it could be wild cattle, or it could be pasture that you have, or the cattle that you have at your house. Mushrooms tend to grow on these. So these are called as coprophilous fungi. So mushrooms that love growing on dung. Then there is something called as entomopathogenic fungi. Okay, so these are mushrooms or fungi that parasitizes insects and grow on them. Then you have mycorrhizal mushrooms, or mycorrhizal fungi, which have symbiotic relationship with. Can anyone guess? Myco meaning? What is the term myco mean? Okay, myco is something related to fungi. Rhiza is related to. Okay, rhiza related to roots. Yes. So these are relationships between the fungal mycelium and roots. So certain mushrooms can have relationship with the roots of trees. Then there are mushrooms that can parasitize another other mushrooms. So that is the last mode of nutrition. Okay, so decomposing wood or plant matter, live tree, fertile soil, coprophilous fungi, entomopathogenic fungi, mycorrhizal fungi, and parasitic, or mushrooms parasitic to other mushrooms. So here you can see mushrooms that are growing on live tree and dead and decaying plant matter. Okay, so these are few of these examples of this particular type of mushrooms. Now here, if you see, both of these mushrooms, if you see, are growing on fertile soil. So if you find an inocybe, okay, you will always find it in, or it growing on fertile soil. You will not find an inocybe growing on tree. Okay. Similarly, you will not find an auricularia growing on fertile soil. So uh, uh, okay. So these are, so they are specific. They have specific preferences. So can anyone guess what mode of nutrition this is? These two. Can anyone tell? Yes.
Leaf dead and decaying
No, these two,
saprophytic condition.
yes, but they are coprophilous fungi. Okay, they are coprophilous fungi. So both of these were found. No, they were not growing on elephant dung. So these were growing on cow dung. Okay, so this was from a region which was adjacent to wilderness. So you will see a lot of, yeah, you can see a lot of fiber in this. Yeah. Then what is this? Can anyone guess what this is?
Is that a root, sir?
No, it's not a root. It's related to the question. So there was a question above regarding the Last of Us, right? Yeah. So this is a Ophiocordyceps. So it is growing on an insect. Can you all see the insect?
Yes, sir.
Yeah. So usually you will find a lot of these growing on cicada. So cicada, which are so most of the times
We
the
can also
insect will
see
be
them on
underground.
caterpillar heads, right, sir?
I didn't get the question. Can you repeat?
We can also see
এগুলো
some
আমার
type of
লেটনা পিছফালে
parasites
ওলায়
on caterpillar head, right, sir?
Yeah. So that you will find in the Himalayan foothills.
We saw
The
that in Uttarakhand, sir, in the
Yes.
FRS.
So that is the Ophiocordyceps sinensis. So that is used in Chinese medicine. So this is an Ophiocordyceps, different Ophiocordyceps species. Now there are many cordyceps and Ophiocordyceps species that grow on cicada. There are genera of fungi that grow on spiders. So there is something called as a gibellula, 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 that usually grows on arachnids. Okay, so based on again, they are they have host specificity. Okay, that is a entomopathogenic fungi. Then you have this particular mushroom is a mycorrhizal fungi. So you won't be able to see the relationship here or with your eyes, but based on continuous observations, you will be able to understand. So based on the type of trees that are usually associated with this particular mushrooms, when you find it, you'll get to know this particular organism. So this image was taken from Uttarakhand. This mushroom was growing in a sal forest. So I assume that it is mycorrhizal. It has a mycorrhizal relationship with the sal trees. Because I know Amanita mushrooms, many Amanita mushrooms are mycorrhizal, especially this particular section of Amanita, that is Amanita section vaginae. Then there is something called as, so this is, what is this? Can anyone guess? The last type of substrate. No, it's not a button mushroom. It's not charcoal. So this is mushroom growing or parasitizing another mushroom. So that's a mushroom parasitizing another mushroom. So the black mass you see is actually a blackening Russula. So Russula is a mushroom
Parasite.
genus. Okay. So the white thing that you see is an Asterophora, which is another mushroom. So Asterophora usually parasitize these particular Russulaceae members. Okay, Russulaceae members and grow from them. So yeah, you can find mushrooms in weird substrates also. So these are few images of mushrooms that like when they started acting weird, right? So again, it all depends on the enzymes that the mushrooms can produce. So if the enzyme that the mushroom produces can digest the food or the substrate, it will grow on it. Okay, so the substrate specificity is usually a what you say, it is based on what enzyme the mushrooms can produce or the fungus can produce. Okay. So substrate was the first one. So substrates are very important because they can be specific, right? Mushrooms can have specific substrates. There are also possibilities for a single mushroom or a single species to have two different substrates. Again, it is based on the enzymes that they produce. Types of mushrooms. So this is based on the overall stature or overall structure of the mushroom. So the terms that I have mentioned here are generic terms. They are not scientific terminologies. So these are terms that you can use to describe a particular mushroom in your normal or in your day-to-day conversations. First one is an agaric mushroom. Okay. So most of the mushrooms, I would say, most of the mushrooms that have gills on the underside. So gills. So gills are basically radially arranged sheet-like structures. Okay. Radially arranged sheet-like structures. So these radially arranged sheet-like structures are called as gills. So most of the mushrooms that have a gill, which have a stipe and the cap. So that is a agaric type of mushroom. So agaric type of mushrooms, the underside will usually be gills. Okay, you can see the image here. Okay, so the button mushroom that you eat belongs to Agaricus genus. Okay, genus Agaricus. So this agaric term comes from genus Agaricus because, as I said, when only macromorphology was the tool, when microscopy was not yet introduced into the taxonomy of mushrooms, most of the gilled mushrooms were classified under genus Agaricus. So whatever species or genera that we have now, most of them were under Agaricus. So that is where the term comes from. So this is a pluteous mushroom. Then the second type of mushroom is a bolete mushroom. So boletes are mushrooms that have the cap, same way how the agarics have. But on the underside, they have pores. So they have pores on the underside. They don't have gills, they have pores. So the spores are produced and released through these pores, and they have a stipe. They are usually very fleshy. So very fleshy in the sense they are very spongy. So yeah, so the one you see on the right is called as a Tylopilus. So here I have mentioned the name. It is also called as a bitter bolete because if you do a taste test, so you take a small piece of it, nibble a little, and if you spit it, you will feel the bitterness. So usually they fall under boletails. Okay, boletails. The order boletails comprises of boletes. Then you have something called as polypores. Okay, so polypores again, they don't have a cap, stipe, or the general, the usual mushroom structure that you see. Instead, they have a shelf-like structure that is present on tree. Okay, so they grow on tree. Usually, they grow on tree, and they'll have a shelf-like structure, and you will see poroid underside, pores on the underside. Just like in boletes, they form or they produce spores and release through this underside. Yes, these are also called as bracket fungi. They are also called as bracket fungi. Then there are gasteroid fungi. Okay, so gaster, what does gaster mean? So I've written there. Yeah. So gaster is basically, it means a sac-like structure. So gaster basically means a sac-like structure. So in this particular type of mushrooms, the immature spores are usually enclosed within a sac-like structure. So here the white mass that you see here is the immature spore, and the you can see the outer layer. So that is why they are enclosed in a sac-like structure. So if you are into, or if you have experience in documenting mushrooms, you might have come across puff balls, or they are basically ball-like structures. And when you cut open this particular structure, you will find a white mass inside, which has somewhat like a paneer-like consistency. So the white mass is the immature spore. Then you might have also come across particular phallic structured mushrooms. So they are usually they fall under Fallaceae family. Okay, Fallaceae family. So yeah, they are also called as stinkhorns. Okay, stinkhorns. Here you can see one stinkhorn, and you can see another stinkhorn here. This is a mutinous species. It's a mutinous species. Okay. How do stinkhorns spread their spores? So stinkhorns, as the name suggests, they have a stinky exudate. Okay, so the immature spores are usually formed in an egg-like structure like this. And when they start maturing, what will happen? The egg breaks and this particular structure comes out. And on the tip of this structure, you will find a very stinky matrix. Okay, so this stinky matrix encloses the spores, mature spores, and this particular matrix attracts insect. So they attract insect, and the insect are the carriers for the spore. So that is how they spread the spores. Then you have something called as a jelly fungi. Okay, jelly fungi, they have a very jelly-like consistency. They don't have many, what is a specialized structures. So yeah, here you can see three different species of uh, jelly fungi. So this is a fan-shaped jelly fungus. Fan-shaped jelly fungus. And this is a snow fungus. Okay, snow fungus. And this is a Auricularia delicata. Auricularia delicata. So these are some of the jelly fungi. Uh, then you have corals and clubs. So they are terrestrial corals, I would say. Okay, terrestrial corals. They are not aquatic, so they are fungi basically. Yeah, say they assume a coral-like structure. Okay, they are called as coral fungi. They're also called as clavarioid fungi. Again, why? Because there is a genus called as Clavaria. So earlier, most of these coral fungi were classified under Clavaria. So that is why it is called as a clavarioid. It is also called as clavarioid fungi. Okay. Now, the next thing that you should note is the location. Okay, so where you found this particular mushroom from. Okay. Now, certain mushrooms can have a global distribution and certain mushrooms can be endemic to certain regions. Okay. Now, uh, some mushrooms like marasmius and coprinella. So, if you can see here, the marasmius is usually, so this data is again from iNaturalist. It is a research grade data, uh, or the research grade observations. And but again, you are not finding much observations from these particular areas, but they are found there. So don't get me wrong that they are not found there. They are found there, but again, you will see a wider distribution, right? So they have a wider distribution. Now, if you see Copernilla section domestici, again, they have a very wide distribution. Now, there are mushrooms like Termitomyces. Okay, Termitomyces is a genus of mushroom that is only found in Asia and Africa. They are a type of mushrooms that have symbiotic relationship with termites. Okay, so they have certain relationship with termites, and these particular mushrooms are only found in Asia and Africa. Okay, they are only reported from Asia and Africa. They are edible mushrooms, one of the tastiest mushrooms. During monsoon, you will find a lot of these being over harvested. Actually, some of these people go around, they destroy the termite mounds to take these particular mushrooms. So again, that is one reason why you should not go around and collect all the mushrooms that you see without having a prior knowledge. Okay. Then this particular mushroom, the Auriculoscypha anacardicola, this was only described from Kerala, or only what do you say, reported from Kerala. But again, there are reports from Karnataka also. So they usually grow on, or they can be found growing on mango trees and cashew trees. So you can see the species epithet, right? Anacardicola, which is related to the Anacardiaceae family trees. Okay. So yes, so distribution. So that is why location is very important. So whenever you are uploading an image, you have to specify the
Sir, can
location,
I have
because
that location slide one? I need
I
to take
didn't
a
get you.
screenshot. Sir, can I have that location slide one? I need to take a screenshot of.
Oh, 1 second. Yeah. So this one actually it is reported from Kerala
Yes, sir.
and
Thank you.
Karnataka. Okay. Now the next type is a type of growth or next characteristic. So some mushrooms usually grow solitary, or they grow alone, but some mushrooms can grow in scattered groups, and some mushrooms you will find in large clusters. Again, this is something, this is a very species specific characteristics. Okay, it's a species specific characteristic. Uh, yes, it is reported from Maharashtra also. It was, I think, yeah, this year it was reported if I remember correctly. Uh, yeah, solitary, scattered groups, and clusters. This information is also very important because certain mushrooms grow solitary. Okay, they don't usually grow in groups. And certain mushrooms like this fairy ink caps you will find in large clusters. Okay, so that information is also very important. Shape of cap. So we have covered the general characteristics or the characteristics that are not the morphological characteristic, but usually it could be the environmental conditions or all those things. We have covered that, and we have come towards the. So we are starting at the cap of the mushroom. Okay. Now the mushroom can have different cap shapes. Okay. It can be plain or planar. It can be convex. It can be pulvinate. So pulvinate is basically a semi-spherical type of cap. It's a semi-spherical type of cap. Then you can have a egg-shaped or a ovoid type of cap. You can have a cylindrical type of cap, a conical cap. So there is something called as a campanulate. Okay, so a campanulate type of cap is basically bell-shaped cap. So it is a bell-shaped cap. You can have an umbonate. Umbo is a slight bulge on top of or on the tip of the cap is called as a umbo. 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 So cap or mushrooms that possess an umbo is called as a umbonate type of cap. Then you can have an umblicate. So you have umbo, which is a small nipple-like structure. So it is a small elevated structure, and umblicate is a depressed structure. Okay, it's a slightly depressed structure. So morphologically, it is usually described as a navel-like structure. It's a navel-like structure. Then you have a larger depression here. So that is a depressed cap. Then you have something like a funnel shape. So this is called as an infundibuliform. Infundibuliform. So that is a funnel shape. In colloquial or in a normal layman's term, you can call it as a funnel shaped mushroom. Now, sorry, this is not convex. It is a concave shape. I'll type it down in the chat. It's not a convex. It is a concave shape. Concave shape meaning a shell-like shape. So usually the oyster mushrooms that you get from the market is a concave shaped mushroom. Uh, Again, different colors, textures. You will find a lot of these in different species of mushrooms. You can see in these particular images. So you can see here a different structure, sorry, texture to this particular mushroom. This one is called as a fiber cap, again, an inocybe. So the cap, if you see, it is very fibrous. So these are certain observations that you can note down. So one important characteristic is presence and absence of vellum. Can anyone guess what this vellum is, or these powdery structures that you see on top of the cap? So these brown spots. No, they are not spores, but they are remnants of universal veil. So we spoke about universal veil. So when the universal veil persists on the cap, you will call them as vellum or velar particle. Vellum. VELUM. Okay, that is a vellum. So you might have seen the emoji in WhatsApp, right? The red and white. So the white spotted red mushroom, right? So that is an Amanita muscaria mushroom. So the white spots that you usually see are actually remnants of a universal veil. So they are remnants of universal veil. Then again, here you can see different sizes and shapes. and different textures. So this is called as an echinoderma. Okay, echinoderma, you might have heard it in another
Animal
taxa.
kingdom.
Yes. So echinoderma meaning, what is the meaning of echinoderma?
Echinodes means like brittle,
Echinos meaning spiny,
something like that.
very
Yeah,
rough.
fine.
Yeah. So echinoderma meaning spiny skin. Yes, spiny skin. So echinoderma, they usually have this particular texture. So this is what I meant by the concate shape. So here you can see the pleurotus or the oyster mushroom. So this is a oyster mushroom that was growing on a mango tree. Yeah, it was a naturally occurring pleurotus. So it's not a cultivated one. Then underside of a mushroom, as I said, what is the function of underside? Anyone? Okay. So the function of underside is to, yes, produce spores. Yes, it's for producing spores. Gills for breathing is in fishes, not mushrooms. So in mushrooms, it is a reproductive structure. It is a fertile surface. So some mushrooms can have gills on the underside. So where you have sheet-like structure. So if you take few pieces of uh, paper and if you arrange radially around a stalk-like structure, you will get something which is similar to this particular what do you say, gills. Here you can see something called as a false gill. Okay, so earlier in gills I said you take a piece of paper, right? So they are different pieces of paper that are arranged radially. But in case of what you say, false gills or this particular structure, they are not actually gills. So instead of taking, let's say, so so gills, it will be sheets of structures like this, right? But in case of false gill, it will be just foldings like this. Okay, it will be foldings of the skin. So that is the difference between the false gill and a true gill. So they are also called as ridges. So they are just folds on the skin of the mushroom. Uh, then you have poroid underside, porous underside. So this is again a hexagonia mushroom. So hexagonal pores you will be able to see. And you will also find certain mushrooms having teeth on the underside. Okay, so these are called as hydanoid fungi. Hydanoid fungi. Okay, so mushrooms can have four types of underside. First one is gills. Second one, so gills usually found in agaric type of mushrooms. Poroid underside you will find in bolides and polypores. Bolides and polypores. False gills usually you will find in certain mushrooms called as cantharellus. Okay. If you are into this culinary mushrooms and these particular foraging or these activities, you might have heard of chanterelles. So chanterelles are very highly priced mushrooms. So they are again foraged mushrooms. Okay, so they people consume them. They are a delicacy. So they usually have this particular ridges or false gills. That is one of the major characteristics that most amateur foragers use. Okay, we'll go through few of these questions. So if I have missed any questions, please let me know. Any do comment on the fungi on frog case that became big news last year. Yes, so there was a mycena that was growing on frog. So from the Mala forest, that specimen was from Mala forest. So again, mushrooms are or fungi are continuously evolving. They always try to adapt for or try to evolve towards a better substrate or new substrates, right? Again, the reason behind that particular phenomenon was like we couldn't, or we don't really know much about it right now. Any law which governs sales of commercially cultivated and native wild mushrooms in India, both for consumption and cyclic ones, also wild ones with no known uses. So, any collection of any wild mushrooms from a forest area is an offense. So, you cannot collect from there. So, if it is growing in your backyard, if it is naturally growing, you can collect it. It's not an issue. The edible ones, not the psychedelic ones. Psychedelic ones, again, the psychedelic mushrooms as such is not listed under the list of narcotic substances, but you will get caught if you do such activity. So better to stay away from that. Uh, sales of commercially cultivated. I don't think there are any such rules for cultivated culinary mushrooms or medicinal mushrooms. Why are some mushrooms in color and why most of them are in white color? I have seen actually most of the mushrooms are not white in color. Like there are like many mushrooms that are colored. Color of cap is a identification characteristic also. So bright colors in animal kingdom you will usually associate it with toxicity, right? But in case of mushrooms, it's not that case. Most of the toxic mushrooms are usually white in color. So if you see the Amanita phalloidae, okay, Amanita section phalloidae, it is a toxic section of genus Amanita. You will find a lot of these mushrooms are white in color. In contrast to that, the edible genera or edible sections in Amanita, like Amanita section Caesare, are brightly colored. So that particular rule is not applicable in case of mushrooms. And why do mushrooms have colors? I am not sure why. I don't know if there. I don't know if there is a reason for it. Reason for the color. I don't know if it serves any evolutionary advantage for the mushroom. Because it's not always the same color, right? There are like many different types of or different colored mushrooms. How do syngons that have okay? How do clavirate and jelly fungi spread their spores? So they usually produce the spores on their surface, and it could be through wind or through rain or the water. They can spread their spores. If it is at the bottom, how will it reproduce? So I don't think it matters much, because yes, if it was taller it would at least get transported by wind. So in case of most fungi, the spores are actually transported by wind. So there is a study that shows that mushrooms produce their own wind current. So that studies are also there, wherein the mushrooms, based on the differences in their charge, electric charges, they produce a wind like or a air current which pushes the spores towards the upper side. And many mushrooms grow on a elevated surface. Okay, so mushrooms that usually grow on trees usually grow on an elevated surface. So that is also there.
So I have a question.
Yeah.
So how exactly are spores produced? Are they produced within the mycelium network, or are they produced within the fruiting body? And
So
how are they produced in that case? Like
okay, so to understand this, you have to go to the cellular level again. So these mushrooms on the gills, they have certain structures called as basidia. Okay, so the basidia will be producing or will be producing the spores, basidiospores. Okay, so basidia is basically a club-like cell that is present on the gill surface. And this particular, so these are basically specialized cells. Okay, these are specialized cells that produce the spores. So usually at the tip of this particular club-like structure, you will find It could be four spores present at the tip of this basidia. Uh, so yeah, based on the species, right? So that is how they are formed. Any other doubt? Uh, what is the function of false gills? So false gills, again, it is the functionality of this particular false gill is same as that of the true gills. Functionally, they are not different, but morphologically, they are different. Are truffles found in India? One second. There was a question about truffles above. What are truffles? So truffles are mushrooms or fungi that usually grow below the soil. Okay, so they are growing below the soil. Uh, so they come under ascomycota, I believe. So they are not under the basidiomycota that we usually, or the usual mushrooms that you usually discuss about or you find in nature. So they are in the ascomycota. Uh, Okay, truffles found in India. I'm not sure about that. I believe they are found. Within the same species, can fruiting bodies have different color variants? Yes, there are color variants. So they are VARs, VAR, VAR. So there are many VARs for certain mushrooms. So yes, there can be color variants commonly or uncommonly noticed during foraging. Yes, we'll. So if you practice this particular, what do you say, documentation of mushrooms, you will come across this particular, what do you say, variance or color variance. If mushrooms exhaust its substrate, will it die? So in case of mushrooms, they usually propagate and they usually spread in search of new substrates or in search of better substrates. Okay, so once the nutrients, once the nutrients are deplenished, so what will happen is they, yes, the mushrooms will be malnourished and they will eventually die, but again the mushrooms will continuously produce spores and these spores will be falling on a newer substrate and it will start colonizing that substrate. So it, I don't know if the, if it you can rightly say death in case of mushrooms. Yeah. Okay, how can we identify mushroom families? Again, identification is basically based on this collection of characteristics. So I have discussed few of these characteristics. I think I'll continue with this and I'll take the questions at the end. And if you have any doubts, you can always reach out to me. Uh, yeah. So color of gills, again, color of gills is an indicator of the color of spores. Okay. So a mushroom that, so here you can see a salmon pink color spore. So usually the mushrooms that belong to, uh, what you say, Plutiaceae family have this particular color on their underside of the mature mushroom because of the color of the spores. Then you have mushroom like Gymnopilus, which is called as a rust gill. Again, the color of the underside is because of the color of the spores. So the spores which are deposited on the underside or which are produced on the underside stains the underside of the mushroom. This is why image of an underside of a mature specimen of mushroom is important for identification because the spore color is sometimes specific to certain genera. For example, as I said, the this particular color of spores is found in Plutiaceae family. Plutiaceae family. Again, here you can see an immature agaricus, which is having a pink gills. Okay, so in case of agaricus, the gill color turns from white to pink and pink to brown. Pink to brown. Here you can see a dark brown. Then there are mushrooms like Chlorophyllum molybdites, again, green spored parasol. So it is a green spored mushroom. Here, Lactarius indigo is a white spored mushroom, but the underside itself has a blue color. So that is why it is visible as blue color on the underside. Mm. Okay, gill arrangement. Again, the length of the gills. So here you can see all the gills are equal in length, right? So it starts from the margin, ends at the stipe. Here you can see there are certain smaller gills in between. So this is called as an unequal gill. So an equal gill, unequal gill, and there is something called as an anastomosing gill, wherein you will find vein-like interconnections between the gills. So this is called as an anastomosing gills. Now, why these are important? Because Russula usually have a equal gill. Okay, most of the Russula have equal gill. So this particular informations are very important when you come to a conclusion, right? When you have to conclude an ID. Then another thing that you should document is a how the gills and stipes are attached. Okay, how the gill is attached to the stipe. Here you can see something called as a free gill, wherein the gills do not touch the stipe. So here you can see there is a zone of free zone, right? This is called as an adnexed gill. So it will gills will touch the starting of the stipe, starting of the stipe. And here you can see an adnate gill, wherein the gills will uh, touch the stipe, but towards the lower part. Okay, so that is called as an adnate gill. And you have something called as an decurrent gills. Okay, so there is a decurrent gill wherein the gills will run along the stipe. It will run along the stipe. Okay, so these are different gill attachments. You can collect my number from Ashwin. Uh, Ashwin, please. Okay. Then again, we discussed about partial veil, right? So these are the partial veil remnants that I was talking about. Now, in some mushrooms, it can be absent. In some mushrooms, it may form a ring or a skirt like structure. Okay, a ring or a skirt like structure, like this in agaricus. And here you can see a membranous structure, right? So these are certain structures that you should document. Again, and these characteristics are specific to certain genera or species, presence or absence of these particular, what do you say, ring or skirt like structures on the stipe. Now we'll move towards the base of the stipe. So we started discussion from the cap. Then we saw the gill attachment to the stipe. Then we saw the stipe. Then we are at the base of the stipe. Now some mushrooms can have a bulbous base. So what is a bulb?
Like onion.
Yes. So bulbous is base is basically a enlargement towards the base of the mushroom. So here in this particular mushroom, if you see, you will see this particular structure clearly, right? So that is called as a bulbous base of the stipe. Now here also you can see it's slightly bulging outward. So that is called as a bulbous base. So this is an Agaricus abruptibulbus. Abruptibulbus is a cotyledonous species. Many cotyledonous mushrooms can have this particular bulbous base. So yeah, documenting this is very important. Then there are certain structures. So again, can someone guess what this structure is? What could be this? Volva. Volva. Hmm? Yes, it's the universal veil remnant. So it forms a cup-like structure on the underside. So that is the vulva. So certain genera like Volvariella, Volvopluteus, and many Amanita species will have this particular vulva. Then there is something called as a pseudorhiza or a false root-like structure that some mushrooms can have, not all mushrooms. So Termitomyces is one genus that predominantly have this particular structure, pseudorhiza. Pseudo means false, rhiza meaning root. Some mushrooms can also have visible mycelium at the base. Okay, there is a mycelial structure at the base of the mushroom. uh, yeah, mushrooms can have certain bruising or bleeding reactions. So these are based on what do you say, oxidative reactions. Okay, now you have to check if the mushroom that you have, you can slightly damage or slightly score the stipe or the cap and to see the bruising reaction. So that is something that you have to check. Or some mushrooms can bleed or form certain secretions. Okay, here you can see. And again, you have to get the cross section of a mushroom to identify. So, in case of gasteroid fungi, puffballs, you have to take a cross section of the mushroom for identification because from outside they look similar, but when you cut it, you will find that the inside of these particular mushrooms can have different characteristics. The last characteristic is the spore print. So, which is actually it is not a necessary step, but it could help in getting the spore color. Okay. So basically, what you will do is you will take the cap of the mushroom, you will place it on a paper or an aluminum foil, put a drop of water on top for the moisture, and you will close the cap with a say a container. Okay, so that there is less air current. Now, after few hours, say overnight, you can keep this, and if you take the cap, you will see that there is a print of spore that is formed. So something like this. So that is basically a print formed by the spores. So these are few spore prints that I took some years back. So you can see the spore colors very clearly. And if you have a microscope, you can view the spores under the microscope. Now, certain tips for documentation of mushrooms. So, whatever characteristics that we discussed till now, you have to document these characteristics. So, for documentation of such characteristics, you have to take the image of mushroom in its natural habitat, where so in its natural habitat undisturbed. Then you should click clear images of underside. So in case if you are not if you find it difficult to pick a mushroom, you find it ethically difficult to pick a mushroom, you can place a mirror at the base of the cap, and you can click images and share. Okay. And in some cases, a cross section like this will be very important because to see the gill attachment. Then clear images from different angles, basically top view, side view, the underside images, the cross section. So these are all very important. So the entire mushroom showing the base of the mushroom is also important. Then the location and date of collection is important. Information about substrate. So even though we'll be able to see the substrate here, it would be better if you can include that information in the description. Then note on staining or bleeding. If you observe any staining or bleeding when you cut the mushroom, that is also very important. Then the type of trees that are growing around. Again, if the mushroom is suspected to be a mycorrhizal mushroom, these informations are very important. Then smell of mushroom. So many people, it is difficult to identify the smell of a mushroom. So again, that is not a important characteristic. But again, if you can get the smell, it would be helpful. Now, few points to note while uploading mushroom observations in INAT. So you have to upload clear images as mentioned earlier. You have to add location, then include these informations, whatever we what do you say mentioned earlier in the documentation of fungi. Avoid duplication. So this particular trend that I have been seeing, especially during the city nature challenge, is that images will be taken from different angles, and these different images will be uploaded as different observations. So that is not the proper way of identifying or proper way of documenting these mushrooms and uploading in iNaturalist. The different angles or different images that you take, club them into a single observation and upload it so that identification is also easier. Do not add IDs suggested by iNaturalist if you are unsure. Again, in using macromorphology, most cases you will be able to go till the genus level for identification of mushrooms. So if someone uploads a mushroom image and gives a ID to the species level, it will be difficult for the person who is identifying to disagree to it because they are unsure. Because the person who is identifying is also unsure about the species level ID, right? So it is always better to leave it at the uh, leave it at a level where you are comfortable with. So if you leave it at fungi, that would be good, because again, people who are into this fungi and mushrooms, they usually filter their observations with kingdom fungi so that we get all the observations. So again, that is a good option. Uh, don't leave it without an ID. That again will be a problem. Then you can mention these accounts for ID. Okay, so these are few accounts that you can mention. Indian Toadstool is my account. Wombat is Vyom Bhatt. So he is again a friend of ours. And Epicurean Jinu, we are all, so we three of us are admins of Mushrooms of India. We co-admin along with other admins. Then Mushroom Manny. So this is Manuel Miguel Mendoza. He is from North America, and Alan Rockefeller. So these two are foreign identifiers. They are again very important part of this community. And these are few accounts that you can mention for identification of mushrooms. And in case if you find an entomopathogenic fungi, this is the person you have to go for. Rick Tehen or Richard Tehen.
Oh.
He's a researcher who works on entomopathogenic fungi. Yeah. I think the session got a little extended beyond our expectation. Uh, yeah.
Thank you, Hari. I think it was a great session. And more or less all the questions have been answered. So if there are any other questions, maybe you can just still put it in the chat box so that Krishnan can answer it.
Is there any truth in the belief mushroom attracts snakes, and another belief like rain accompanied with lightning? So the first question, the belief of mushroom attracting snakes is not true. That is not the case. But in many places, I have heard about this. There are many misconceptions in when it comes to mushrooms. So there are other beliefs like if you pick a mushroom and put it in turmeric water, and if the color of the water changes, the mushroom is toxic. So there is no golden rule in identifying edible and toxic mushrooms. There is no golden rule. If someone say that there is, it is wrong, it is false, because you cannot identify a toxic mushroom based on a single characteristic. So some people say that mushrooms that grow on tree are non-toxic. But again, the funeral bell or the Gallerina mushroom, Gallerina marginata, grows on wood, which is a deadly mushroom. So there are no rules when it comes to mushrooms. Accompanied by lightning will grow more mushrooms next day. So there is some science behind this particular relationship of lightning and mushrooms. One is some studies show that the the sound waves help in the growth of mushrooms. Another study shows that the mushrooms or the lightning can fix nitrogen. So nitrogen is essential for the growth of mushrooms. So again, these are some of the like we can't conclude from these studies, but there is some data that backs this particular claim. And again, we have been observing this from a long time. Are all mushrooms that grow on termite mound belong to termitomyces? Are no, all mushrooms that grow on termite mound are not termitomyces. There are some silaria that have relationship with termites. So again, termitomyces, identification of termitomyces, you can go for presence or absence of pseudorhiza. So pseudorhiza is again a good indicator of termitomyces. Uh, if you are in doubt, get it identified from someone who is good at identification without jumping into conclusions and consuming any mushroom. So before consuming any mushrooms, you should make it a practice that it gets identified. What is the substance in mushroom that makes it toxic? So there can be different chemical components. So it can be amatoxin. So there are different toxins. So in case of amanita phalloidae group, it is amatoxin. So there are toxins like muscarin. So all these particular toxins or chemical compounds that interact with our body. Okay, so when I say a mushroom, when I say that a mushroom is toxic, it is toxic to humans. So you should understand that other animals can consume it. Okay, so that is one reason why you should not eat a mushroom that another animal is eating. Okay, you can't conclude a mushroom is safe if some other animal is eating. Squirrels can eat one of the most or some of the most toxic mushrooms. Okay, so they are immune to these toxins, but these toxins or these chemicals can interact with our body and it can induce certain adverse effects. So that is how we, or that is why we call them as a poisonous mushroom or a toxic mushroom. Uh, Reliable resources for learning about more about fungi. So if you are looking into, so again, fungi, it could be, so again, mycology have different aspects. It can be identification, cultivation, medicinal mushrooms. So all these different, what do you say, streams or branches are there. So if you, so if you tell me which is the way you have to go, like which is the path you want to choose, I could guide you to people who might be an expert in that. Okay. So yeah, I think I can take those in like you can DM me or chat, send a message to me. What's the difference between pseudorhiza and chlorhiza? Okay, so uh, here if you see the chlorhiza that I had mentioned is not the structure. Okay, it is the genus name. Okay, so it's not the structure name, it's the genus name. So chlorhiza ambonata have a pseudorhiza. So these also have pseudorhiza. So contact details. If you are in Instagram, you can contact me at Indian Toadstool, and this is my phone number. Right. Okay, this is my personal number. You can yeah, message me. I heard plucked oyster mushrooms can generate vitamin D when exposed to sunlight. Yes. So mushrooms or fungi are more related to animals than to plants, or we or humans are more related to fungi than we are related to plants. Okay, so most of the cases when we say that we have the ability to what do you say, synthesize or generate vitamin D. Similar way, fungi or mushrooms also have this ability. So you will get these sun-dried mushrooms in market, which is actually mushrooms that are fortified or mushrooms that are exposed to sunlight, which accumulate this vitamin D or which generate the vitamin D. So consuming these is a very good source of vitamin D. It can be considered as a good source of vitamin D. Any other doubts? I think I've answered most of the questions. If you have more doubts, you can always reach out to me. Uh, yeah.
I think there are no more questions.
Yeah.
Harikrishnan. So thank you very much. Indeed, a very interesting session because I don't think we can do many other things like birds, frogs, amphibian, reptile, but fungus is something I don't think, at least I have never heard a session on this, and this was a fantastic one. Thank you very much for doing this session. And I hope everybody else has also thoroughly enjoyed and understood this. So like Harikrishnan has said that please use the right identification on the INET, and City Nature Challenge is just down the road this coming weekends. A lot of excitement happening everywhere. So 25th to 28th, please remember to participate. And this
Yes.
session will be uploaded onto the Citizen Science YouTube channel. So you'll get the session recording also over there. So thank you very much, Harikrishnan, once again.
Thank you. Thank you so much for giving me this opportunity to present here. And I think I was part of the CNC last time also. I think, yeah.
Yes.
So, yeah, it's always nice to be a part of this.
And I think you'll get many more tags now that you have shared your email, your phone number, and also
Yes, yes.
Instagram ID. So
Yeah.
thank you very much, and good night, everyone.
I'm always happy to receive questions and doubts on mushrooms.
Fantastic.
Yeah.
Thank you so much for the support.
Thank
Okay.
you. Thank you.
Very
So
much.
see you all.
Good
Bye.
night.
Good
Bye.
night. Good night.
I'll end the meeting now.
Yeah.